Hello, everybody, and welcome to this CRISPR Medicine News webinar. My name is Karen O'Hanlon-Cohrt. I am Editor-in-Chief at CRISPR Medicine News, and I am delighted to welcome you to today's webinar on the topic of disrupting the care of cardiovascular disease with single-course gene editing medicines. Our speaker for today is Andrew Bellinger, MD, Ph.D., who is the Chief Scientific Officer and Chief Medical Officer at Verve Therapeutics. Dr. Andrew Bellinger is the Chief Medical Officer at Verve Therapeutics, a cardiologist and scientist who specializes in company creation and growth, building high-performing research and development teams, translational medicine drug delivery, and has a particular interest in the clinical development of novel classes of therapies. Prior to joining Verve, Andrew co-founded and served as Chief Scientific Officer of Bellinger Therapeutics, and prior to that, he was the Chief Scientific Officer of Cocoon Biotech. He is also a co-founder of Cohrt Therapeutics. He previously practiced as an attending cardiologist at Brigham and Women's Hospital and is board certified in cardiovascular medicine and internal medicine. Andrew holds an MD, a PhD, and an MPhil from Columbia University, an MS in mathematics from New York University, and an AB in physics from Princeton University. He completed his clinical training in internal medicine at the University of California, San Francisco, and his clinical training in cardiology at Brigham and Women's Hospital. He completed postdoctoral research training in drug delivery with Dr. Robert Langer at the Massachusetts Institute of Technology. If you have a question for Andrew, please type it into the questions box on your screen, and we will get to the questions after the presentation. That's it from me. Over to you now, Andrew. I look forward to hearing your presentation.
Well, thank you, Karen. I'm delighted to be here and have the opportunity to share a little bit of the story of Verve Therapeutics and how we're seeking to disrupt the care of cardiovascular disease. Forward-looking statements, standard disclosure. Atherosclerotic cardiovascular disease is the #1 cause of death worldwide. And that's despite a number of available treatments. I think we all know this in the abstract, but it's critically important that people, I think, understand the huge impact that cardiovascular disease continues to have in the world today. One person dies every 34 seconds from cardiovascular disease in the United States alone, and there are hundreds of millions of people worldwide who suffer with this disease. Over 800,000 heart attacks per year in the United States are attributed to this disease. This is, we understand ASCVD fairly well, the biology. It's due to high cumulative lifelong exposure to blood cholesterol that builds up in your heart arteries. And those cholesterol come in 3 different forms: low density lipoproteins, LDL particles, triglyceride-rich lipoproteins, and Lp(a). The solution is known. It is to keep your blood cholesterol as low as possible for as long as possible. This comes from extensive human genetics work, as well as from prior studies of other lipid-lowering therapies that have shown convincingly a relationship that low LDL cholesterol for a long period of time will reduce or eliminate your potential for developing heart disease over a lifetime. But why doesn't it happen? Well, there are 3 pills and 3 injections that are available today to lower cholesterol. What's the unmet need? The reality is that in the case of ASCVD, only about 50% of patients in the US are on any lipid-lowering therapy, and only about 27% of ASCVD patients reach the current LDL goal established by the American Heart Association. Only 27% reach goal. The problem in heterozygous familial hypercholesterolemia, a genetic subset of ASCVD on the right, is even worse. Only about 3% of patients are reaching treatment goals for lipid lowering, despite the availability of these 3 pills and 3 injections to lower LDL cholesterol. This is from a global registry of heterozygous FH patients. What's the reason? We believe at Verve that the fundamental challenge is that the chronic care model to treat chronic disease is broken. It sounds obvious that for a chronic disease like ASCVD, you simply need to take medicine every day for the rest of your life. But the reality is that the vast majority of patients do not want or succeed in achieving this goal. Daily pills and intermittent injections that need to be administered over decades impose a heavy burden on patients, on providers, and on the healthcare system. And the fact is that it rarely happens. At Verve, we're focused on two forms of genetic subsets of ASCVD. These are patients with familial hypercholesterolemia who have sky-high LDL from birth that leads to development of ASCVD at young ages. There are two forms of this. On the top in blue is heterozygous familial hypercholesterolemia. This is typically caused by a single copy mutation in a gene such as the LDL receptor, which leads to LDL cholesterols in the range of 190 and above milligrams per deciliter. Patients with heterozygous FH often develop heart attacks in their 30s, 40s, and 50s. And there are about 3 million such patients in the US and Europe who carry this diagnosis, who have this diagnosis today. There is a much rarer form of homozygous familial hypercholesterolemia that is present in about 3,000 people in the US and Europe. This is where you have mutations in both copies of the LDL receptor, leading to sky-high LDL, often over 500 milligrams per deciliter, that starts in early childhood. And the vast majority of these patients do not reach LDL goal despite being on 4 or even 5 lipid-lowering therapies. So our pipeline at Verve is designed to address this problem. A pipeline of one-time gene editing medicines to disrupt the chronic care model and come up with durable, permanent solutions to lower LDL cholesterol starting earlier in life. Our lead programs are against PCSK9. This is a highly validated gene that we'll talk about in a minute. And the initial indication is heterozygous familial hypercholesterolemia for the PCSK9 program. We have 2 products against that target, Verve 101 and Verve 102, and we'll talk about both of those. Our second program is against ANGPTL3, another gene that where turning off the gene in the liver can lower both LDL cholesterol and triglycerides. And this target is particularly attractive for the homozygous familial hypercholesterolemia patients, where there is an approved antibody, evinacumab, that targets this gene in that patient population that's on the market today and very successful. Our third program is against Lp(a), and this is in patients with genetically high Lp(a) from birth. We have 2 other programs against undisclosed targets. So I'm going to start with focusing on Verve 101, our lead program targeting PCSK9. This is an adenine base editor in the form of an mRNA and a guide RNA targeting PCSK9 that are encapsulated in a lipid nanoparticle. The lipid nanoparticle is shown in the 4 components of the lipid nanoparticle are shown in the middle, and the form on the right. This takes a form that is very similar to a COVID vaccine in that it has this similar 4-component lipid nanoparticle structure encapsulating an mRNA, but this one goes to the liver. How it works is shown here. It's a lipid nanoparticle is a one-time infusion into the blood that goes to the liver. At the liver, it's taken up by LDL receptors on the hepatocytes and internalized and delivers the mRNA and the base editor and the guide into the cell. The adenine base editor goes to the nucleus where it finds the single base pair in PCSK9, the adenine, that is at a splice donor at the end of exon 1 in the gene. And it turns that adenine into a guanine. And in doing so, it disrupts splicing and turns off expression of PCSK9. It can do this at both copies of the PCSK9 gene to reduce the production of PCSK9 from the liver and then subsequently lower LDL cholesterol in the blood. Preclinically, we've extensively evaluated this program, and shown here is one such study, a fairly large study in non-human primates, 36 non-human primates in 3 groups, a vehicle group and Verve 101 at 2 dose levels. And you can see that Verve 101 in non-human primates lowers PCSK9 by about 80% at the higher dose, and durably for beyond a year and a half here. And on the right, you can see the LDL-C cholesterol reductions range from about 50% up to upwards of 70% at the higher dose. For comparison, inclisiran, the siRNA targeting PCSK9, can lower LDL-C cholesterol by about 40% in patients with heterozygous FH and about 50% in patients with ASCVD. So we think this has the potential to be as potent or better than inclisiran, with the potential for durability that could extend for the lifetime of the animal. With that data, we initiated a Heart-1 phase 1b clinical trial last year. We began dosing patients in July of 2022. This is a single ascending dose study design where we test about 4 dose levels with small cohorts of 3 to 6 participants in each cohort. The study was initiated in New Zealand and in the UK, and we are working to expand the country footprint beyond that. The recruitment is ongoing in both New Zealand and in the UK, enrolling patients who have heterozygous FH, as well as several additional high-risk features, including ASCVD and an LDL-C that is not at goal on standard of care. We are guiding towards data in the 4th quarter of this year in 2023, and that will include both safety as well as pharmacodynamic data on PCSK9 and LDL-C reductions in the cohorts as we progress through the study. Overall, our plan for this program is to establish proof of concept in these high-risk heterozygous FH patients, and then expand into all heterozygous FH, which is about 3 million people in the US and Europe, into a phase 2 and ultimately a pivotal phase 3 study with an LDL-C reduction endpoint. The broader vision of the company is that one day this could be a treatment for the broader group of ASCVD patients who have high lifetime risk for coronary disease. And that is estimated at north of 50 million people in US and Europe. We have a second program against the PCSK9 target. This is Verve 102, and it represents an evolution on Verve 101 with a number of potential modifications. It uses the same editing approach of an adenine base editor against the same site in PCSK9. However, the lipid nanoparticle is a GalNAc lipid nanoparticle. So it's a 5-component lipid nanoparticle using a different ionizable lipid licensed from Novartis, and with a targeting agent called a GalNAc lipid that allows it to get into the liver cells through an alternate mechanism of uptake. And you can see this particle on the right. The mechanism is similar, but the delivery, the lipid nanoparticle delivery is a little different. So it's still an intravenous infusion that goes to the liver, but now it gets taken up by 2 different receptors in the hepatocytes, both the LDL receptor and the ASGPR receptor that GalNAc is a ligand for. And this allows it to bypass LDL receptor, bypass the LDL receptor, which is particularly important in some patients who lack LDL receptor. The editing machinery is identical to Verve 101, and the result will be a similar reduction in PCSK9 and LDL-C. Preclinically, we've shown similar levels of potency of Verve 102 in wild-type non-human primates. And the plan is to initiate a phase 1 trial of Verve 102 in the first half of 2024. This is a program that we initiated over 2 years ago and have been progressing through IND-enabling studies and CTA-enabling studies through the past 18 months. Our second program targets ANGPTL3, and this program will also be initiating a clinical trial in the second half of 2024. ANGPTL3 is a different gene that is also associated with where inactivation of ANGPTL3 is associated with both lower LDL-C and lower triglycerides. So the human genetics, which was actually done by one of our founders, Sek Katharison, our CEO, identified that patients who lack ANGPTL3 completely have remarkably lower LDL-C and triglycerides. Shown here is a woman, Anna Fior, who is a human knockout of ANGPTL3 and has LDL-C levels of 37 and triglyceride levels of 19. What has been shown in larger genetic studies is that deficiency in ANGPTL3 has no safety effects associated with it, but it does lower blood cholesterol markedly. And based on these data, it has been progressed into a different modality, a monoclonal antibody from Regeneron, which is FDA approved for homozygous FH. The unmet need here in homozygous FH is severe. This is an orphan disease where the vast majority of patients require many LDL-lowering treatments, and even so, few reach goal. Shown here on the left is that even on upwards of 5 lipid-lowering therapies, about half of patients with homozygous FH still do not reach treatment goals. The mechanism has been clinically validated by evinacumab, where LDL-C lowering can be upwards of 50% in these patients in homozygous FH. Our program here is very similar. It uses the GalNAc LNP, which allows us to bypass the LDL receptor and uses the same similar adenine base editing, but now targeting a guide RNA targeting the ANGPTL3 gene. The GalNAc LNP is a proprietary LNP that we developed at Verve, which we believe has the potential to be a best-in-class agent for delivery of genetic medicines to the liver. And this is because of both the specificity of using the acl-glycoprotein receptor and also the potency that GalNAc can provide in getting very efficient uptake and delivery of genetic medicines to the liver. Shown here is a cool experiment where we demonstrated that these GalNAc LNPs, Verve 201, can get into the liver in non-human primates that lack LDL receptor completely. So what we did here is we created a non-human primate model of homozygous FH, patients who lack the LDL receptor. And you can see that at the beginning, we took patients and we actually knocked out the LDL receptor from their liver. And what happened was their LDL cholesterol went from 50 or so at baseline to almost 500 milligrams per deciliter. So this is exactly the phenotype of homozygous FH. And then we came back a few months later and we treated them with Verve 201, the monkey version of Verve 201. And we were able to lower LDL cholesterol in these monkeys that had LDL cholesterol of almost 500 by 46%, exactly matching the therapeutic effect that evinacumab, the monoclonal antibody against ANGPTL3, can have. Our third program targets a third pillar of lipoprotein risk, which is LPA. This is an earlier stage program that is still in preclinical discovery. The high levels of lipoprotein a are genetically associated with risk for ASCVD. The lipoprotein a is another form of circulating LDL particle that is liver derived. and associates with higher risk of myocardial infarction, ischemic stroke, atherosclerotic stenosis in peripheral vasculature, as well as aortic valve stenosis. It is a large addressable market that is distinct from the typical ASCVD patients that just have high LDL cholesterol from, say, heterozygous FH. So about 20% of people have Lp(a) levels greater than 150 nanomoles per liter, about 11 million patients in the US and Europe. And these are distinct from those who have what many would call garden variety ASCVD risk factors like high LDL cholesterol. It's a natural potential situation where a one-time gene editing therapy could be attractive. There are humans who genetically lack Lp(a), and they are both resistant to heart attack and do not have any safety signals associated with absence of Lp(a). The blood levels are very constant over time. They're almost entirely determined by genetic inheritance. There's no alternative lifestyle approaches to lowering Lp(a). And other therapies like statins have minimal impact on blood Lp(a). So we are in the state, in research stages of developing a Lp(a) approach. And I just use this slide to kind of highlight the distinctness of the patient populations that we're targeting with these different approaches. Heterozygous FH in purple is our primary indication for Verve 101 and Verve 102. Homozygous FH, the little red dot, the very small orphan condition, is the primary target for our ANGPTL3 program. And high Lp(a) in green is a distinct patient population that is targeted by our third program. So, I think a question that people often ask is, why one and done therapies? And I think it fundamentally comes down to those of us who are clinicians and take care of these patients see this day after day. The pattern of care for most of these patients looks something like this. Patients are undertreated early in life with high circulating LDL or other lipid risk factors, and only initiate treatment following a clinical event like a heart attack. The standard of care, lifestyle changes, statin medicines are effective, and they do lower LDL cholesterol. Addition of a PCSK9 monoclonal antibody can be even more effective at helping people reach goal. However, the majority of patients 2 years after their heart attack, well, more than 50%, are not taking those therapies still. And the reasons for that vary. Often it's that people are feeling well and they stop taking their medicine. Sometimes they are lost to follow-up. They miss a doctor's appointment, or the difficulties of chronic adherence to a therapy lead them to stop. The consequence is shown in the upper right, which is further interventions like coronary stenting procedures, bypass surgeries, and fatal heart attack. Our vision is to change this model by developing durable treatment options that can manage the 20, 30, potentially 40 years of treatment that patients with this disease really need. And take this graph from this to this, a durable lifetime benefit for patients that reduces their subsequent risk for clinical events and fundamentally changes how we think about the disease of atherosclerotic cardiovascular disease. So with that, I'd like to stop and open it up to questions, Karen.
Thank you, Andrew. That was really interesting. Not only a great overview of the work that Verve is doing, but also a great illustration of the massive problem of cardiovascular disease and the various types of diseases. I think a lot of people just tend to think of everything as a heart attack and cholesterol, but there are many different groups in there. And as well, the major shortfalls of the current treatment landscape. To me, it sounds like a no-brainer to go for a one and done therapy and cure the problem, get to the root cause. I suppose the cholesterol reducing medications are among some of the most widely prescribed medications worldwide, and probably also a massive source of revenue for those who develop them. So I suppose I'd like to start, before I open it up to the floor, by asking a couple of questions about how expensive a base editor therapy would be in comparison. And I mean, how easy do you think it will be to get this shift that you're looking for in the care? I mean, governments would need to ultimately fund these treatments for people. So what do you say about that?
Yeah, what we're trying to tackle is a big problem. It is, millions of patients. It's a different situation than a lot of the other gene therapies or even other gene editing approaches where they're targeting very small patient populations. And what that means is that to be commercially successful, we don't need to charge millions of dollars for each treatment. And in fact, the beauty of the approach that we've taken, not using a virus to deliver not using an AAV or other virus to deliver the genetic medicine, is that there is a potential for a relatively cost-effective therapy. And the reason I say that is, I mentioned this early on, but the lipid nanoparticle encapsulating an mRNA for an adenine base editor approach that we're taking is structurally and from a manufacturing standpoint very similar to a COVID vaccine. And we know from the COVID vaccines over the last 3 years that those can be manufactured very cost effectively at a low price point. So what this means is that there is a path to developing this therapy in a cost-effective manner that could be available to the millions of patients who actually need it and suffer from this disease. So that is something that we are, from a vision standpoint, committed to, that we make this in a cost-effective manner.
Right. Yeah, no, that sounds really, really positive and optimistic, especially with the lessons we've learned from the COVID vaccines. Do you actually see base editing therapies for these indications replacing the current standard of care, or do you foresee a future where both exist still?
Yeah, I think for the foreseeable future, this is about an additive therapy on top of existing standard of care in terms of lifestyle modifications and statin, perhaps ezetimibe. For a given patient, I think the decision will ultimately be which treatment option do they want, which treatment option is right for them. Would they rather take a monoclonal antibody injection that they self-administer every 2 weeks at home? Would they rather come into the doctor's office and see their cardiologist every 6 months to get an intramuscular injection like inclisiran? Or would they rather get a one-time intervention? And our conviction, our belief, is that a meaningful fraction of patients and providers will decide that, for a 45-year-old patient who had a first heart attack, that patient is looking at 35 years on average of lipid lowering therapy. And we know that for that kind of duration, the current treatment options are unlikely to achieve consistent LDL-C lowering over 35 years. And for a patient like that, we think that a one-time gene editing therapy has the potential to really truly be a superior approach, not just an improvement in convenience or something like that.
Yeah, no, that makes a lot of sense. So you're developing Verve 101 and then Verve 102, which are very similar, but as I understand it, the difference is mainly the delivery strategy. So the difference of that one can be taken up by 2 receptors, the 102. So Verve 101 seems to be working well so far. If Verve 102 goes into the clinic and also works really well, do you think the company will plan to commercialize both?
The clinical development programs right now are small phase 1 studies. But as we get later into development, they'll become much larger studies. And our approach here is that we want to we want to develop the best product to take forward into these larger indications. So our plan is to evaluate both Verve 101 and Verve 102, essentially side by side in phase 1 clinical studies, to determine which one is going to have the best potential to be advanced into those late stage clinical development programs that are much larger and much more expensive. So I think the plan is to evaluate them, compare efficacy, safety, dose levels, and then determine which one is the right therapy that has the best potential to reach the broad problem that we're trying to tackle here in ASCVD.
Sure. So you mentioned that we can expect data readout on the 101 trial in the last quarter of this year. Are you able to tell us how many patients have been dosed so far?
Yeah, we haven't given kind of play-by-play of each patient dosed, but we have guided that we anticipate having enough data to share about the ascending dose response to give visibility into the dose response.
Okay, so one of our attendees is asking if you can elaborate on the dose levels in the phase 1 trial, but perhaps you can't say any more than you've already said.
Yeah, the approach to a single ascending dose study like this is to start at a dose that a starting dose that is established by having a wide safety margin from the doses used in your tox studies, and then increasing the dose up into the therapeutic range. So that was the approach that we presented to regulators and have taken forward into the clinic.
Yeah. Thank you. So far, the programs are each targeting one gene. We have somebody in the audience here asking, would there be any advantage to base editing multiple gene targets together as a cocktail for any of the patient populations?
Yeah, it's a great idea, and in fact, one that we have piloted. We presented at JP Morgan, I believe, in 2 years ago in 2022, a non-human primate study where we encapsulated in the same lipid nanoparticle guide RNAs targeting both PCSK9 and ANGPTL3. And when we did that, we were able to simultaneously, with a single injection, turn off both genes. So one of the beauties of base editing is that unlike Cas9, the kind of first generation double-stranded break cutting, you can target 2 genes simultaneously because you're not cutting the DNA, you're just making single base pair changes. And so the risk of introducing 2 cuts in the DNA and having translocation events is much, much, much lower with the base editing approach. So we have piloted it. It does work. And I think the question is, is it an attractive regulatory path to take a combination, dual target editing strategy forward? And in which patients would you do that? Homozygous FH is a natural choice because those patients, the majority of them do end up on both a PCSK9 and an ANGPTL3 therapy. And certainly, targeting both genes has the potential to take somebody with an LDL cholesterol of 400 all the way down close to the treatment goal just with one therapy. So, that it is a very attractive idea and one that we do continue to consider. I think we're developing a very new modality here. It's a lot of novelty in what we're doing, and that applies certainly on the regulatory side as well. And we're trying to take a very staged approach to that, to develop proof of concept that we can safely base edit both PCSK9 and ANGPTL3 individually, and then I think we can talk about those kind of other approaches.
Yeah, right. We will watch out for further updates. Am I right in saying that Verve's 101 that is the only in vivo base edited program to have been dosed into a patient so far? Am I right in saying that?
In vivo base editing, yes. As you know, I think Intellia has demonstrated some really impressive data with traditional Cas9
right.
approaches in vivo as well.
Yeah. So let's just talk about delivery for a minute. So we've hosted many delivery-focused webinars in the past. It's obviously, it's a big challenge. Maybe it's less of a challenge for the liver than some of the other organs, but you've mentioned the GalNAc LNP your proprietary GalNAc LNP approach. So somebody is asking about biodistribution. So it goes to the liver. Where else does it go, or does it go anywhere else? And have there been any concerns about off-target editing in those tissues?
Yeah, our team presented some data at the European Society of Cardiology meeting just last week in Amsterdam on Verve 201, which uses the GalNAc lipid nanoparticle. And what we see is that the biodistribution is really pretty tightly restricted to the liver. And that's true in both mice and in non-human primates. There is a little bit of biodistribution to a few other organs like spleen and adrenal gland, but it is much, much, much less than what goes to the liver. So it is a fairly specific mechanism of delivery for a gene editing medicine, which is important because, of course, you don't want editing activity in non-target cell types if you can avoid it.
Yeah. And another question related to delivery from our audience here as well. Do you see non-viral delivery as the future?
Personally, yes. I think certainly for indications like this that have larger patient populations and broader broader use cases, I think non-viral delivery is likely to be the right approach for a variety of reasons, certainly safety being one, but also cost. It would be very difficult to be cost competitive with existing treatment options in atherosclerotic cardiovascular disease using a viral approach.
Okay, thank you. So still quite a few more questions, actually. It's been a really interesting talk, a lot of nice comments. I won't read them all out, but a lot of excellent talks here coming up in the comments. So have a follow-up on the cocktail question. Somebody is asking if you think there would be an advantage to using alternative Cas proteins in a cocktail.
Our approach to each target, we're a cardiovascular focused company. We're not a technology focused company. So we select our targets based on the human genetics, based on the previous pharmacologic evidence. And once we select our target, then we evaluate different technologies. So, for our lead programs, base editing is the best approach for PCSK9 and ANGPTL3. And we determine that by comparative evaluation of other approaches like Cas9 or like other novel Cas enzymes. But for each target, you have to make that assessment. And for some targets, base editing may not be the right approach. For some targets, Cas9 may not be the right approach. And you may want to use other editing technologies, other editing enzymes. And that's something that we're actively working on for some of our discovery stage programs. So I think the answer, is a qualified yes, that there are at times there are going to be other editing approaches and other editing enzymes that are better suited to that particular genetic target.
Would you consider split dosing in such cases?
Yeah, split dosing is something that we've demonstrated it preclinically in non-human primates. We've incorporated it into our tox studies from the get-go. We do think, this is about providing lifetime treatment. And if your goal is lifetime treatment, then whether the treatment is one infusion or 2 infusions is, I think, less relevant than the durability of the treatment. We believe our approach is likely to be durable for decades, if not a lifetime, based on the preclinical data we have and others have about how the liver turns over. When we make a DNA edit, it is a permanent edit in that cell. And as liver cells turn over, they carry and propagate that same DNA edit to their progeny. So we think this is going to be a lifetime edit for people's liver. And if that's the case, then doing split dosing is very, very reasonable, and may actually, improve the kind of therapeutic index of your that may be an optimal dosing regimen. Sure,
thanks for that. I'm almost finished with the questions. I was thinking about the homozygous familial hypercholesterolemia that is more prevalent in children, or at least in one of your early slides, you had some numbers on children who have this disease. And I was wondering how easy is it to spot the children who have this disease? How early can, somebody receive this treatment in their life? And would this require some rollout of screening as well to find the people who would benefit early on in life?
Yeah. In the case of homozygous FH, these patients have typically such sky-high LDL cholesterol that they actually can have manifestations even in early childhood, age 5, 6, 7, 8. And that can include things like cholesterol buildups on their elbows and wrists. Xanthomas is what they're called. And so homozygous FH is actually pretty well diagnosed today. Some patients do slip through, and there is a push towards more pediatric screening for high cholesterol. Most national societies around the world are recommending a one-time lipid measurement in children between the ages of 10 and 12 to measure both LDL cholesterol and potentially in the future Lp(a). But a lot of that screening, Karen, is directed towards heterozygous FH, actually, which is severely underdiagnosed in children and undertreated as well. I think, coming to kind of our treatment approach, in homozygous FH, we will likely pursue a kind of adolescent pediatric approach at some point in the future, given that many of those patients do have pretty severe manifestations of disease. They can have heart attacks in their, in their latency years and early teenage years. So I think we will likely pursue a kind of adolescent pediatric strategy for homozygous FH. For heterozygous FH, we are focusing on adults who can, consent themselves for a one-time genetic therapy, which, I think for the foreseeable future really, can be restricted to adults.
Yeah, it's also a much larger group, so a greater chance to impact straight away. The last two questions then, I suppose one really, the people who would get such a treatment, do they still have to watch their diet?
There's a lot of benefits to watching your diet. It's not just about lowering your LDL cholesterol. It's about, managing your weight, your physical fitness, your ability to, maintain a high activity level throughout your lifetime. So as a cardiologist, I strongly recommend everyone stay active and, live a healthy life, because that you're going to achieve a lot of benefits from that beyond LDL cholesterol lowering.
Yeah. And the last question, so it's clear that Verve is a cardiovascular company. So, I assume you're not going beyond cardiovascular indications right now. Many of the programs that are currently in your focus area can be tackled by base editors, by fixing or creating single point mutations. Are there any major cardiovascular diseases that Verve would like to go after, but maybe can't do it with a base editor? Do you see Verve taking in other types of gene editors as necessary to expand the indications?
Yes. We do actually, on our pipeline, we have one program that's partnered. So our one program on the pipeline is partnered with Vertex, and that is not a cardiovascular target. So we do have one program, but that's a program that ultimately will be, the clinical development will be led by Vertex. So our cardiovascular focus is really for our wholly owned programs and our programs that we're driving the clinical development of. And I think that just reflects that, that is, we think it's a very large problem. And it's one that we think we are uniquely well suited, given the team that we've built with expertise in cardiovascular disease. With respect to technology, we truly are technology flexible. We are focused on the disease and on the patients. So there are we have existing programs that where we think that we will use alternative technologies to base editing. And we have a pretty active program in evaluating different editing technologies against some of these targets beyond the first two, which are committed to base editing.
Okay, great. Thank you. That was all my questions. Can you take one more from the audience and then we'll wrap it up?
Sure.
It's just this one is kind of interesting. It comes up a little bit when we speak about trials, gene editing. This person is asking what strategies are in place to persuade patients to embrace these therapies, given their one-time and irreversible nature. Do you have any comments on that? Has it been any issue?
It's a great question. I think what we're developing is a very new modality, and that understandably means that there's a lot of education involved. And that's the primary approach that we take to talking with patients and, through our investigators is education. We educate them on the treatment options today. We educate them on their disease course, and, what base editing has the potential for, as well as the risks. What we've found is that the motivations for a lot of participants come down fundamentally to this being a familial disease. So I would say, two archetypes would be perhaps, let's imagine a 45-year-old person whose one of their parents died at age 50 from a heart attack. They know they have a familial disease that puts them at high risk. That can be very motivating for people to try an alternative approach. The other archetype is perhaps a little bit older patient who has 3 children, 2 of whom have been diagnosed with heterozygous FH. For them, it's about moving science forward to make new treatment options that could provide more durable lifetime benefits to their children. And that can be very motivating as well. So I think, at the end of the day, it's all about patient autonomy and about informed consent and informed decision-making. And that's the approach we take, is to, educate and offer options. And I think, what we found is that there are a lot of patients who are looking for other treatment options. And we think that the approach will resonate, particularly as we have more and more data that we can share publicly.
Great. Yeah. Hopefully you'll come back and talk to us in the future again when you have more to share about the data. I think we'll wrap it up there, if that's okay with you, unless you have anything you'd like to add.
No, I just want to say thank you. It's been a lot of fun.
Yeah, thank you so much for the presentation and a brilliant discussion. I also personally really enjoyed that myself. So that brings us to the end of today's webinar. Many thanks to Dr. Andrew Bellinger once again for taking the time to share his knowledge with all of us. I wish you and Verve and all the patients the very best of luck with the therapies going forward. We'll certainly be watching out for updates and publishing them as they emerge into the public domain. And finally, I'd like to thank our audience for listening in and taking part in the discussion. And we hope to see you at a future CRISPR Medicine News webinar. So take care.
Thank you.
Bye.